by now, you know that we're going to be dealing with neurons. And so all of that stuff, right? So we have these different approaches, computational neuroscience and neural computation, but all of them depart from the neuron. But not both of them see the neurons in all of this degree of complexity. This is actually a micrography from the brain where they have used a particular dye called the brainbow, brainbow, that colors the neurons in different colors, depending on their protein makeup. But the one thing that I wanted to share with you from this picture is how packed neurons are in the brain. We often think about them as these abstractions here, but in the brain, you actually have tissue, and tissue from the brain comes from the skin. Evolutionarily, we're not going to talk about that so much, but it's something that you might go back to and find it out. Neural tissue is an offspring of skin tissue, so it's continuous in that respect. Dendrites came later. Spikes are what neuron produces, and now you've heard the word a couple of times, and the basics of it, everything got from secondary school. You inject current to the neuron, and the neuron spikes. I guess that this is clear to everybody. This is a representation of a patch clamp, things that we're going to be discussing tomorrow. So how do you inject current on a neuron? How do you make a neuron spike? We're going to be discussing this tomorrow. So here you have some steps, current injected, and here is a spike, but here it produced an all or non-spike. So this is the textbook, secondary degree textbook vision of what the neuron is. But now we know that the neuron, neurons produce way more different patterns that are not always simply an on or off response as a function of certain input. Some neurons spike all by themselves. So these are neural activities, different categories. We're going to be breaking this slide down in exquisite detail in one later lecture. But for the moment, I just want to impress upon you the fact that the real brain produces loads of different types of spikes. And this is then the material that we have to begin with when we're thinking about computational neuroscience and neural computation. The last part of the lecture today is trying to make this distinction clearer. This is a slide to summarize what I've been indicating when we were talking about the mind map a moment ago. Computational neuroscience and neural computation view neurons and networks in slightly different ways, but they have different levels of modeling, one would like to say. One more accurate, a lot more less. Abstraction here, low level and high level, might disagree on. This is the way that we generally talk about low level and high level. Low level is looking at the detail with a strong microscope. High level is looking at the distance. Focus here is on reproducing experiments, on measuring stuff from biology, neurobiology, testing hypotheses that are physical in nature often. When does the neuron produce a spike? We're looking into biophysics, that is capacitance, conductance. We're looking at the physical counterparts of neuronal activity. We talk about plasticity, which is that neurons change, that synapses change as an opposition to learning. Right? So computational neuroscience is way more concerned with how exactly do neurons change than how they change to acquire certain things. And here our focus is in anatomy, whereas here our focus is in circuit. The abstraction that allows us to talk about how our brains organized to do something. For example, if you hammer this knee, my leg will kick. There is a very simple circuit that explains that, right? That you've also learned about in school. It's negative feedback with the forward trigger. It's a circuit, and we also made many circuits. Some simpler than others. Some of them are so messy that it's hard to tell apart. Nevertheless, for neural computation, the interest would be what are the motifs that evolution is searching for as it's trying to organize the brain. Whereas here, we're actually concerned with the specifics of anatomy. How is brain connected? Alessandra Badura will tell you a lot about this. The language, therefore, here is closer to the language of physics or neurodynamics. The details matter a lot. What's the ion channel, specific protein? Dimensions is in the term of, if you want to make a model that represents this, the neurons here tend to have many dimensions in the sense that you're encoding a lot of detail. And here you're encoding less detail in the neurons. A abstract model of the neuron is nothing but a circle that is sending inputs and has few internal variables, few internal dimensions. This is an abstraction that will become clear as we discuss more for the next 10 weeks. I'll talk about dimensions quite a bit and we will discuss what they mean. They also have a couple of cards related to them. The computational cost here per unit, that's the asterisk, is large. If you want to compute a single neuron at its exquisite detail, might take weeks. Whereas in computational neural computation, Google will compute millions of neurons right now on that chip. But these neurons are much simpler. So the computational cost per unit is larger in computational neuroscience. Questions often revolve the very quantitative. How much electrical field will we observe when we see a synchrony of these many neurons in the population? Here is whence does pattern recognition come from? Whence does memory, right? So where does it come from? Why this particular circuit arrangement is good for this? It's more of a qualitative take on the entire question. So the bottom line, is this is the thing to remember, and is on this side of the dashed line, we have simulation of neurons, and on this side you have computation with neuron models, things that approximate what we think of neurons are. The dashed line in the middle is lightly dashed. I don't mean to imply that there is a cleave between the, or a big watershed. Often they talk to each other in the middle, but often they don't. To make Google's Alpha 0, plays chess very well, they did not ask any neuroscientists how to play chess. Right? They just engineered the hell out of a neuron-based system with reinforcement learning to do it well. But we would like, here in this course, we're actually traversing this thin line at all times, or at least we will try to do that, at least conceptually. So even though there are these distinctions that help us think about it, the essences are subsumed in these two categories, I would like to think. By the way, what I did here was modeling disciplines with tenuous verbal language. Right, so this is not, I tried to create two categories and explain what they are and how they relate to each other. So this is also a type of modeling, and some people might disagree with my model. Now let me give you a flavor for what we're going to talk about in the course. In computational neuroscience, you're looking into exquisite reconstructions of single neurons. This is a Purkinje cell of the cerebellum that someone looked under a microscope, traced the dendrites, traced like an individual, made little compartments that are electrotonic here, imagining that they are interfacing with the world through some sort of currents that are being exchanged in voltage that is maintained in each one of these segments. Look at the detail, right? So it's hundreds of compartments. And nowadays we do it even more so. Computational neuroscience will sometimes describe individual ion channels on this membrane. Very accurate. Very accurate. Can't be perfectly accurate because we cannot even measure where they are, but then we can make assumptions about how many should be more or less, and see if that will give us explanatory / predictive potential. In computational neuroscience, we're often talking about prediction more than in neural computation. Compare this with the super boring, very easy element of computation in most modern neural networks. You have inputs that are numbers that represent the activity of other neurons. So these are inputs to a neuron. This is a neuron, a ball. And that neuron sums those inputs They maybe have some weights coming in. 1, 2. They sum those inputs together. So what do? They multiply and sum. I mean, how is that like a real neuron? Well, right. So does it abstract something fundamental of the operation of the neuron in that case? Certain case more than others. These are establishing the entire dimension. Okay, so we can discuss how plausible that representation is. And then he has another operation here, which is in that case, this line here symbolizes a sigmoid function, some sort of output function that relates the input to the neuron. So the sum of this input. We're going to talk about this more, but I want to tell you what that little sigma there represents. So if the inputs to that neuron are this much, like say 127 number, it will output a value between 0 and 1. Maybe very active, right? So the representation is that the activity of the neuron here is highly abstracted. We're not talking about voltages. We're talking about a number that represents how active the neuron is. It's not even a firing rate. It's just a number between 0 and 1 that says very, not very, something in between, right? So we've abstracted all biological complexity away, elements of neural computation. The onset of computational neuroscience is in this paper, 1952. Hodgkin and Huxley, Nobel Prize winners, explained where the action potential came from by making a bunch of assumptions about, what the neuron was made of. Tomorrow more about this. This equation produces action potentials. It's a set of differential equations. This dot is dt. So this is dV dt. How much voltage change over time, capacitance, current. We're going to talk about these terms in the project to exquisite amount of detail so that you understand this model fully. So this is one of the goals that I have for this course. Even though it would seem daunting now, I hope that by the time that you finish with the project, you will have a substantial sense of what does it mean to explain the onset of the action potential via this model. It's a flavor, okay? So I'm not going into detail now. It explains the onset of the action potential via some applied currents as the interaction between ion channels in the membrane. How many of you have ever heard about an ion channel? Interesting. And you will learn more about them and then create more representation as they go. Nowadays, we're able to reproduce all of the spiking patterns that we observe in reality via simulation models. On the fly, we can tune them to a degree of 99% of accuracy in certain cases. So we can create a model neuron that to 99% reproduces the outcome of a real neuron given a certain input. It's pretty mind-boggling with simplified models. So this is then what computational neuroscience is dealing with. It's taking the biology, simplifying into some highly abstracted compartments potentially of this neuron to explain the action potential. This is, I'm going to skip this neuron, this slide. This is about compartmental modeling of the neuron. So, but I'm going to skip this thing. Using Hodgkin-Huxley type model neurons, these cosmologists here, no, they're not cosmologists, but it looks like a cosmology paper because there are so many authors. So if you're not really very much into the literature, scientific literature, this should impress you. There's a lot of people working on the reconstruction and simulation of neocortical microcircuitry by Henry Markram and others. This is the Human Brain Project. And they thought that it was not too early to try to describe neurons to exquisite detail and compute them, put them in a simulation model of the brain where each layer has neurons with known branching properties, known ion channel properties, known spiking properties, putting them all together. Oh, so accurate, so complex. What are we missing? Where are the wrong parameters? How much can go wrong? Lovely questions. Can we carry on discussing them? Ultimately, I can tell you that from these, you produce
quite a bit of explanation and predictions. So this is the activity of that column, if it's actually going. Is it going? Good. Yes. So every single neuron here being active is representing an action potential in that neuron. This is slowed down from real time. And what do we learn, right? Oh man, like it's just pretty pictures. Oh man, like you just put like 100 people to work together on characterizing neurons, and what do you get out? Just a video. No, you get actually predictions, right? And it's sometimes incredibly compelling predictions about what the activities of these neurons will be upon novel stimuli, but at a physical level. Because these models are physically constrained, then the kinds of explanations, the kinds of predictions we make are also constrained by the biology we observe. So we're trying to bring them together. It's kind of like, okay, we have a plane and we know it flies, and we start to copy parts of it and see if, so is this a good part? And maybe, if we copy well, then the aerodynamics are already given. And in many cases, because the electrical model of the neuron is not bad, we can get very far with this. I'm not going to go into the details of this, but I am going to say that there's a number of predictions that came out of the circuit from this paper, which are very compelling. Some of which we didn't know about, some of it which we have already tested positively, some of which are wrong, and then we're going to have to work more. So this is the extreme on one side of computational neuroscience, like a full-fledged model of an entire piece of the mouse brain, many neurons. But you don't need to work with those complex models, especially if you want to do something with the neurons. You would like to try to think about what is it that you're trying to do, and what are the essential components for that, what you're trying to do. So it might not be that a gene in that moment. Here's an example. So guy gets an uranium poisoning in Japan, 1960s. Complete uranium poisoning. It has every single nucleus in his brain destroyed within that moment. Okay, he survives for a week. In the first day he's taking protein. So there are no more protein-making machinery in the brain. There's no more cells with nuclei. You look at this data exists, and yet, still to a certain extent, the cognitive functions were processing. So there were elements of this circuit that could abstract away from protein production. Now, not for long, of course, but this already proves a point to a certain extent. So we can then go from a very complex, detailed compartmental model by a reduced compartmental model, making a single compartment model with fewer variables, making a cascade model that goes all the way to a black box model that might look like a ball. And then you take those balls and you try to think, okay, well, how am I going to put them together to do stuff? And the stuff that we're going to do is what we talked about a moment ago when we were saying, cognitive neuroscience includes the functions that we know we perform, the outlines that we might explain via pattern recognition, categorization, object tracking, with your eyes, motion perception, something is moving, decision making, motor control, navigation, pattern generation. All of these are things that you might think that balls can do if appropriately wired, if appropriately thought of. Now, one thing that we need to realize before we even start trying to do that is that everything that we know that we get in from the environment and from the world is exquisitely well within as patterns. Spatial temporal patterns that are coming through us through our senses at all times with a lot of correlation. We're not getting snow, from the TV. We're getting stuff. As I move here, like the visual image moves in specific ways. So these contingencies help us understand how pattern processing will happen in the brain. And so we take that assumption from the outside world. This type of view that I'm espousing here is called embodiedness, right? So you're embodied, and because of that, you're embedded in the universe, and because of that, the patterns that we're processing are structured, statistically regular. So we can, on the basis of these two things, the fact that we know that there are functions in the brain, and the fact that we know that there are patterns that come through our senses, to try to combine them into different architectures that will produce specific functions in neural computation. So this is a sampler of a bunch of different kinds of artificial neural networks that nowadays exist and are being deployed for different purposes. We're going to discuss many of them, amongst many, fakes, deep fakes. Those are generative adversarial networks. They have a certain architecture. And that architecture is an assumption about what circuit does a certain function, okay? So just to get you a sense that it's not anatomy, is someone's thinking about this. There's one more aspect, and I'm almost finished, which is how networks change. So in neural computation, you want learning. So you want the networks to change in ways that are meaningful. One of the primary ways in which people train networks historically and even to this day is supervised learning by backpropagation, gradient descent. We're going to talk about this. The idea here, primary idea is that when you give some input to the network, say cat, you give a cat picture, and you want the output to be, yes. Here, like it says 1 if it's cat and 0 if it's not cat. You use the error of your prediction to update the weights such that when you present cat, you get cat. And you do that via backpropagation. How many of you have heard about backpropagation before? Higher. How many of you have never heard about backpropagation before? Of those that have not heard about backpropagation before, how many of you have ever heard about gradient descent? Okay, so only you don't know about gradient, or you know about gradient descent. You have heard about gradient descent. How many have heard about gradient descent? Okay. So there's a little bit of space there. We are going to describe what backpropagation is, what gradient descent is. This is for supervised learning. In unsupervised learning, networks change by themselves. And we need to have local rules where individual neurons only know what they receive, and the synapses, the connections between the neurons, change as a function of what they listen to. Hebbian learning, for those of you that studied psychology, is an unsupervised learning method. And it can help explain things like the homunculus in the brain. You might know that the brain in sensory systems have like your body encoded in a piece of the brain. And so if you stimulate here, that arm piece in the brain lights up and so forth and so on. These kinds of ideas are represented in unsupervised learning. So the idea of clustering, that a network with the ability to change as a function of activity will absorb the statistical regularities from the input that is given to it. It's an abstract way of saying that the network is changing according to the stimulus. And that's unsupervised learning because it changes by itself. We have rules for the individual neurons. We're going to discuss them. That's in the realm of neural computation. It's in the realm of learning. It's in the realm of function. Last one to last.
My colleagues and I at the University of Waterloo have built a large-scale model of a functioning brain called SPAWN. The model receives its only input as a 28 by 28 pixel image and controls a simulated arm. In this video, we see both the activity of the various brain areas and decodings of the neural representations in those areas. The first task is a recognition task. SPAWN writes the digit it sees in the image. Individual neuron voltage spikes are shown scrolling through the thought bubble at the back of the brain. The decoded value is displayed on top of that activity. The second task is called copy drawing. Instead of just recognizing the digit, the model must reproduce the style of the digit. Here, it successfully copies 2 styles of the number 2. Spawn's representations are thus useful for both categorization and encoding fine perceptual details. The next task is a counting task. This task demonstrates that Spawn can not only represent and categorize numbers, it also has an understanding of numerical concepts like order. Spawn is first shown a number to start counting from, 4, and then it is shown how much to count by, 3. We can see the internal progression of numbers as it counts and finally writes its answer. Next, we have a question answering task. Here, Spawn memorizes a list of numbers and then answers a question about the list. You can watch the topmost working memory as it stores the items in the order they are shown. In this case, Spawn is asked what is in position 5. Spawn answers the question by decoding its working memory and using the result to drive the arm. The final task is a serial working memory task in which Spawn must memorize a list of items in order and then repeat it back. We have seen several tasks that Spawn performs correctly, but the errors it makes are equally important to determining if it is a good model of human cognition. Note that the darkness of the letter in the thought bubble indicates how well that item can be decoded from the current representation. Spawn is now drawing its memory of the list, but when it gets to the 8, it is no longer confident in its own decoding of that memory, so it draws a horizontal line indicating it has no answer.
All right, so just to align on what this model is doing, it was a science paper, by the way, so it's not just a demonstration. It is something that has the devoted effort to create a plausible model of the interactions between the cortex, basal ganglia, and cerebellum to produce behavior, ultimately, right? We're trying. Where is it accurate? It does not produce the same spikes as we were talking about before as in competition with science. But it does give us a good sense that you're closer to how we process patterns to convert them with spikes and with spiking units. This is a spiking unit, but engineered according to theories about how these patterns are processed. So they do have theories. This is a theory-rich approach to brain exploration. They assume certain connections and ways of telling them. And then, because they do and they publish, you can go back to their paper and say, I agree with this, I disagree with this, and we can try to evolve. But there are substantial, we have, this is why I'm teaching this course. So because I think that, this is why I put the minor together, to try to give you a sense that wherever we are right now is an absolutely extraordinary time, where we have the first, steps towards the understanding. There's a lot of stuff to build, and it's still incredibly complex. But there are certain aspects of it that start becoming revealed to us, and through the lenses of a model, we understand more than we did before. Often you will hear someone coming from a more practical science saying, "Oh, we're never going to understand the brain." So it's like, "Well, let's sit together. Let's see what we do understand. Let's see, how much of it can we actually interpret and parse." And this is what the course is about. So for the two fields, consilience is a nice word coined by that guy that says that like when you depart from different points and your predictions start to map onto each other, then you have a truer understanding, to become more confident. So the departure point for both disciplines is the same. Like the neurons are doing stuff, but they're different because their focus is different. So the assumptions about what to include the models, what makes it accurate, what makes it good, differ. But they're both explanatory in their own ways. The explanatory goals differ, but what we're hoping for from all of this is that eventually we will understand what were the detail we had to include, what is the detail that we can abstract away, and how do we step by step assemble the brains. So this is the lecture part of it.